I just wanted to say that normally you save the best presentations for last. But I've seen Reggie speak before. I haven't seen Alisha yet, but I got to see her slides, and I'm telling you, this is a treat. This might be the best session, so I don't know if we could do much better after this, but it's going to be fun. I can tell you that. There's animations and all kinds of things in this. And this kicks off the Cannabis Genomics testing session. And Alisha Holloway is the Director of Bioinformatics with Phylos Bioscience. And she's going to talk to us today about bringing molecular medicine and genomic tools to cannabis. So with that, I'd like to turn it over to Alisha.
Electrons are slow sometimes. Thank you for inviting me here and also for providing early coffee. Thank you to all of you for listening to me talk about the work that I'm doing. I sometimes wake up in the morning and I'm surprised that I can apply the science that I've been trained to do to this amazing field. So I'm working with Phylos Bioscience, and we're a high technology cannabis genomics company. We're using new science to help growers save money, save time, and grow better weed. You all know that cannabis has a long history with humans. We've carried it to all corners of the earth, and we continue to do so. And it's clear why we do that. This plant is possibly the most useful plant that I can think of. We use it for food, fuel, fiber, fabric, pharma. How's that for some alliteration? But really, we've only begun to unlock its potential. It has huge medical possibilities, and in fact, even as a secondary side effect of using it as a medicine, we're seeing drops in opioid uses. This is, as you know, potentially a $25 billion industry over the next few years. And even with the severe restrictions and legal limitations that we have been going through, we've done some amazing things with this plant. You've probably heard of the Iron Law of Prohibition. When alcohol was made illegal, it went underground, it became very potent, even deadly. Luckily, we're not in that scenario with cannabis, but the potency of cannabis in basement breeding operations has gone from 8 to even 30% by dry weight by flower during the time that the prohibition has been in effect. Basement breeding has also produced small plants that may not grow well indoors. So there are a lot of things that have happened to this plant during this basement breeding period. But it's also given us amazing varieties, or cultivars, that have flavor and chemotype profiles that you can hardly imagine. But now that cannabis is legal in many states and a few countries, we can really ask the ultimate question: what can we do with this plant when we bring modern genomic tools together with the decades of knowledge that growers and breeders already have? First, we can and we really have to answer some very, very basic research questions. Answers to these basic questions will help growers, breeders, and consumers. Growers can learn what they have. Is this really sour diesel or something else? Growers can begin to market their products in particular ways so that if we learn about the genetic relationships between cultivars, they can determine whether their cultivar is unique Or if they want to market it more like they do for wine varieties, where you have a terroir approach. Growers can learn what their cultivars are, as I said, related to. Consumers, with some of the developments that we're making, can say, I like Harlequin. How can I get that again? How can I make sure that I'm getting the same thing over and over? And breeders, from the genomic tools that we're developing and studying genetic variation, they can begin to learn what variation exists with the cultivars that we have today. And this is the raw material for selection. So then we can start to apply, once we learn about the variation, we can start to apply modern genomic breeding techniques. We can learn how to select for pest resistance and a robust stature for growing in different conditions that are outside or in large greenhouses. So we've partnered with a few key academic labs to help us answer some of these questions about the biology and genetics of this plant. And we're taking three basic approaches. We're characterizing the genetic variation within cultivars to learn what kind of variation we have to work with. We're learning how different cultivars are related to each other. For example, do all the cultivars that have high CBD cluster together? Are they all related, or do we see that phenotype in multiple clades? And also we can learn how drug cultivars have drawn from natural populations around the world. So we want to integrate all of this information to develop modern genomic tools. We want to map the information to determine whether there's variation for, as I mentioned before, pest resistance or maybe drought tolerance, so that breeders can produce plants that are strong, hardy, and also have even new, unique flavor or phenotype profiles. So to do all the things that we want to do, we need a solid genome assembly of cannabis. We need that so that we can annotate what genes are there, where they are. We can learn about different pathways, the pathways that produce different cannabinoids or terpenes. And we can compare those to model plants like hops that has a genome. And we can begin to develop linkage and physical maps so that we can select for different traits. Now there are existing genomes, there is at least one good existing genome out there. And Phylos has decided to sequence another genome, and why would we do that? Well, we learn a lot from comparing multiple cultivars to each other, the whole genome sequence. We can learn about what's called structural variation, so when you have large-scale rearrangements of the genome. We can also learn about smaller-scale duplication and deletion of genes from one to many or removal of genes. One good example of this is copy number of the THCA synthase and CBDA synthase genes. We know that different copy numbers of those are reflected in cultivars that produce high levels of CBD versus high levels of THC. So we can learn about these pathways by sequencing and understanding the genome. So what are we trying to work with here? Cannabis, plant genomes vary in size. This figure shows, it doesn't matter what the numbers are so much, but it shows that plants, flowering plants in particular, have a huge diversity of genome sizes. So just as a marker here, humans are kind of like mid-range. Arabidopsis, the model plant, is on the smaller side. Human genome is about 20 times larger than Arabidopsis. And cannabis and something like tomato are sort of in between them. The human genome is about 4 times larger than the cannabis genome. So actually we're kind of lucky. Some of these genomes that are really large in plants have lots of repetitive elements and parts that are very difficult to understand and assemble. Also, cannabis has 10 chromosomes, and one of the interesting things about the biology of cannabis is that it has an XY chromosomal sex determination system, just like humans do. And this makes a real difference in the way that you might breed for this. plant compared to other plants that often don't have the chromosomal sex determination system. So here's a sort of busy slide about sequencing the cannabis genome. The Page Lab has done a phenomenal job sequencing the genome of Kush, and they also have a granola resequencing project. They've used what's called short read sequencing, Illumina and 454, so these are short segments of DNA. They've sequenced very, very deeply into the genome and have produced a great draft genome and are continuing to improve it all the time. We felt like we wanted to take a different approach. You know, we still want to be able to compare the genomes to each other, but we used a different sequencing approach. We used Pacific Biosciences technology, which produces these really long reads. And the advantage of that is that you can much easier assemble the genome. So you take these long reads of nucleotides and you put them together, and if the sequences are longer, it's very easy to know where they belong with respect to each other. So with less depth of sequencing, we can get more of the genome assembled. Now we're still working on this. We want to have greater depth of sequencing, and we want to try to cover as much of the genome as we can. So this is still a work in progress. But even though we have just a draft genome, we can start to compare cultivars on a large scale. We can do these things that I mentioned previously. And to do that, we've sampled cannabis cultivars from all over the world. We have covered over 40 countries. We have thousands of samples. And for most of the sequencing that we're doing, we're focusing on a smaller subset of around 3,000 SNPs, or genetic variants. So what have we found? One way to characterize genetic variation is to ask how many sites in the genome are variable. So remember, this is the raw material that we're going to be selecting for to pull different traits out of the plant. And so I put a few other species up here as comparison. Humans, you might say, inhabit this shallow end of the gene variation pool. There's not a lot of genetic variation to work with. I know, maybe we're surprised, maybe not. Soy is similar, but edamame is delicious already. Grape has a fair amount of genetic variation, and in the grape industry there's a lot of concern over pathogens that are taking out some long-lived cultivars that are really important in the industry. It does have a lot of genetic variation, so there's hope that they can use these modern breeding techniques to bring out new varieties as well as find genes for pest resistance. Grape is harder to breed because it's long-lived. It takes 3 years to get to maturity. So we're fortunate with cannabis being an annual. We can move through breeding cycles relatively quickly. And we're fortunate because cannabis has a lot of genetic variation. It has variation that's somewhere between grape and corn. And so we have a lot of material or genetic variation to select on. So I just want to compare corn and cannabis a little bit because it's kind of the poster child of breeding programs. Corn has thousands of inbred lines. That's really different from cannabis, where we really have no inbred lines or not any widely used inbred lines. And so if you take 2 inbred lines of corn, it means that they're within one inbred line, there's relatively no genetic variation. But if you cross 2 together, you get offspring that look exactly alike, and this is the way that corn crosses are done. So they look identical. If you take 2 cannabis cultivars, you get a variety of phenotypes. You have different chemical profiles that vary among the offspring. You have different, possibly colors, statures, yield. And this is actually kind of cool because basement breeders have been allowed to phenohunt using this kind of a crossing scheme. So they'll cross, as you know, pick phenotypes that they like, and then clone them and make lots of cannabis. So now that we can use modern molecular breeding techniques, we can take much better advantage of the variation that already exists in the plant. We can start to stack traits more effectively. And so we can have a robust plant that's resistant to pests, smells like a dream, and reduces anxiety. So that's a little bit about the genetic variation within cannabis. We have a lot overall on the population level, but we also have a lot within each cultivar. There's a lot of genetic variation. And so we wanted to understand how this translates into population structure. Are modern drug cultivars one distinct group of cannabis that's out in the world? How are they related to hemp and other land races? And so we used an approach that looks at population origins or ancestry, a program called Structure. And this is a model-based clustering method that uses the genotype data to infer this population structure and assign each individual that we sample to different populations. So here's an example for humans. So this isn't cannabis, this is humans. Each color on here represents a population. And there are 7 colors to represent the 7 supported populations of humans. Each vertical line on this figure is one human. So if you have a bar that's got some orange, some red, and a little bit of blue, it means that your heritage comes from 3 different populations. This is similar to what you might see in a 23andMe kind of representation of your heritage. So we applied this to cannabis, and we actually have support for 7 populations in cannabis that are represented by these 7 colors. I've named these by some well-known cultivar names or phenotypes. We're just starting to get a sense of really how these populations are what cultivars compose these populations. So the one on the left is OG Kush. It's got members that include OG Kush, White Fire, Platinum OG, Fog. And then if you look, there are, of our thousands of samples, there are a lot that have Kush in their name. And those often will have a large component from this OG Kush population, but they're also a mixture of a lot of different populations. So that word in and of itself doesn't necessarily mean that they're going to be strictly composed of the OG Kush genetic background. And then we have skunk, that includes skunk number 1, Jack Rare, Candy Jack, Jack Flash, things like that. And one of the things that I thought was kind of interesting is that Kim Dog and Sour Diesel have ancestry from OG Kush and skunk primarily. There's the high CBD group. This is one of a couple of areas where we see high CBD. This includes Cannatonic, ACDC, Remedy. We have the thing we're calling berry, which is includes berry white, blackberry pie, black Betty. And then the purples, where we have purple Kush, purple Urkel, God's Gift, Granddaddy Purple. We've also sampled a huge number of what we're terming land races. These are not cultivated in a traditional way like the drug cultivars, but they're from throughout the world, and we have a really good representation from Africa. And these African cultivars fall into this landrace group. We also have a lot from South America and Southeast Asia that also are comprised of this landrace heritage. The final group, and I think that this is not surprising based on other research, is that things that are traditionally known as hemps really do fall out into a separate population group. We have representatives from Russia, Hungary, Brazil, Kazakhstan, Morocco, and so they fall out somewhat separately. One thing that I thought was interesting about the hemp group is that cultivars like Harlequin, C-Rex, and Pitbull have a pretty good amount of heritage from the hemp group. About 20% of their genome is similar to these hemp varieties. So this tells us a little bit about how the current cannabis cultivars are structured. But we wanted to represent this in a way that really compares all of the sample cultivars to each other. So we developed this 3D representation of our data that is what we're calling the Phylos galaxy. So this is based on principal components analysis. Each of the dots that you see, or nodes, in this image, or in this movie, is one sample. And they're colored by those results from the structure analysis. So if you remember, the chems were green, they'll be green in this. And then the lines that connect two nodes together are yet another analysis that tries to infer relationships between cultivars. So which ones are parent-child relationships or clonal relationships. We do have a significant number of samples that appear to be genetically identical. Here's another image that shows you that hemp group a little more clearly. I wish I had zoomed in a little bit more, but there's Finola and the Kazakhstan samples and other things that when people submitted them to us, they termed them hemps. This shows the OG Kush population. You saw at first there were a lot of lines between them. There are a lot of first-degree relationships within this cluster. They aren't necessarily clones of each other. There are some of those, but there's a good representation of this OG Kush group in our data set. Here's another one. So this is interesting. So I want to talk about the names of cultivars. Everyone wants to know, do I have the real Girl Scout cookies? I think that question we're pretty far from actually having a handle on. But some tight clusters of clones have very consistent names. Blue Dream, which comprised, I think, 10% of the market in Washington last year, some large percentage of the market, actually does seem to form a nice tight cluster in our data set. So if you're getting Blue Dream at a dispensary, it's very possible that it's actually what you think it is. Now you'll notice there are a few other names scattered in here. But it's likely that there was a plant called Blue Dream that was cloned and then released to a lot of growers. We also have the converse of this, where you have one name and it's just scattered throughout this galaxy. This is just a final example that I wanted to show you. So I mentioned the purple group. And all of these things you see here in kind of this pinkish purple are clones of each other. But you can see, hopefully, you can read the names a little bit. There is every name in the book in this clonal cluster. There's Granddaddy Purple, which is probably familiar, but then there's Lavender and God's Gift and Purple Kush and Bob's GDP. I mean, almost no name is repeated twice. So clearly, we have a long way to go to clarify what a cultivar name actually means. And in fact, I found out the other day that there are 39,000 named cultivars that were sold in Washington last year. So to help growers and breeders and consumers, we're developing the Phylos genotype test, which you can purchase and send in a little piece of stem to us, and we can put your star on this galaxy. So we've made a fair amount of progress. We're characterizing the genetic variation within the cultivars that we've looked at. We're inferring family relationships. How did different cultivars get to where they are? And now we're starting to work towards developing modern genomic tools for breeders. With the Phylos genotype test, that's a first step, but we have a long way to go. There's really, I think, and we think, a breeding revolution. Cannabis legalization is taking place all over the US and the world, and it's really in parallel with the scientific revolution that's transforming agriculture. More than just cannabis, every cultivated plant. People are using genomic sequence data to breed for all major agricultural crops. And they're using the genomic data to make new plant varieties faster and more accurately than they ever have before. So we want this to be a part of the cannabis industry. We're turning the tools that we're developing into tests so that we can help other breeders with their programs. Breeders can learn about the genetic variation in their lines, know about the relationships between them. We're going to be developing marker-assisted selection methods and using genomic selection. And our goal really is to help breeders bring new iterations of the classics that they've produced to market. So by the end of this decade, breeders will begin releasing new cannabis varieties that we haven't even dreamed of. They're entirely unlike the ones that we have today. I really want to thank you all for listening and thank my team members at Phylos. We are a small startup, so everyone in the company has a role in the science that I've just shown you.